Okay, so we're going to be talking about muscle tissue. And then we're also going to just elaborate on some skeletal muscle like that. Okay, so this is a tennis player, right? So we know that this tennis player, he basically is going to require a whole lot of muscles to sit there and be able to get around and move the way he does on the tennis court, right? And he's going to rely heavily on that skeletal muscles to sit there and provide the force and also take some of the impact of movement. Okay, so let's see. We've already talked about some of the tissues that are involved in muscles, right? Or the types of tissues of muscle tissues, right? Do you guys remember the three types?
Mm-hmm.
Yes, no, maybe?
Yes.
See, I see a text. Yes, skeletal, cardiac, and smooth. And you also remember how to distinguish between them, right?
Yes.
Okay. Okay, so give me some of those features. What is skeletal muscle for?
I believe it's the bone, the holding up of the structure.
Okay. So you're saying it's kind of the holding of the structure. What else do we use it for?
To protect our internal organs.
That's right. It's for protection of our internal organs. Yep. Okay. Cardiac muscle.
The heart. It keeps our heart pumping.
Keeps our heart pumping. Okay. And then smooth muscle. Smooth muscle. Back in the day, I know it's like, man, I don't remember any of that, right? Somebody say something. There we go. Involuntary contractions to help with digestion. Okay, cool. Yep. I also saw stimulation. Good job. Okay, so you want to make sure that you guys remember that for this section. So now we're going to actually start talking about how these muscles are kind of their function itself and how they function. More so the process. Okay, if I can get there. Come on now. So an overview, a quick overview. In this chapter, we're going to be talking about actin and myosin. Actin and myosin are two proteins structures that are involved in muscle contraction, and their organization kind of helps explain how some of the structures are going to be formed, and so it gives us kind of a clear distinction. Like so, for instance, in the case of skeletal muscles, right? We have like a very clear, distinct organization of the actin and myosin genes, and so in terms of that contraction and stuff, that it's going to play a huge role in that. And so the other thing is you know that calcium ion, for those who are taking chemistry, Ca2 plus, or you'll see it in your book written as Ca plus plus, right? So the calcium ion is also involved in that contraction as well. So if you don't have certain ions in your body, you're not able to sit there and perform naturally or the way that. And so calcium is going to be necessary for the activation of certain enzymes, and particularly in this case, myosin. I told you myosin. And then the other one is actin. And so you're going to have actin and myosin chains throughout the body. That's going to be important for muscle contraction. And other components that you're going to be paying attention to is energy. What is cellular energy in our body? Anybody remember? ATP, that's right. Okay. Okay. Clear. Okay, so a couple other things that we want to be familiar with are certain terms. Excitability. Okay, so because, let's see, how can I say this? Okay, so we've talked about that positive charge in terms of calcium, right? It has that positive ion. So when we think about positive and negative, those are charges. And stimulations of your muscles are a result of those charges, right? So positive and negative charges, they send impulses down our system to make it excitable, right? So the plasma membrane of muscles, because they need to be stimulated, are charged up. And so they're going to have charges that are going to be associated with that plasma membrane on one side of the plasma membrane or the other side of the plasma membrane. Okay. And they're going to have what we call electrical states associated with it. Okay. And so that excitability is a result of the change in your electrical state. Okay. Okay. So, so how many of you guys have any of you guys had muscle injury or a leg injury or anything like that, and had to go to a physical therapist?
No.
No. Nobody? Okay. Have anybody tried to lose weight and by using yeah, I guess lose weight by using electrodes attached to your abs? Okay, that's good to know. So next time what I need to do is in your lab kits, I need to put a battery and one of those little electrodes, right? So then you guys can sit there and attach it to yourself and see the result. Okay, so there's these little machines that basically sends electrical pulses through you, right? And so you'll attach an electrode to like a muscle or a region that you need to stimulate, right? And so by us putting that electrode, it causes the muscle to tense up because you're sending electricity through it. So you're polarizing that area, right? And then it goes, and then it stops the electricity, and then it depolarizes. And so you're going to have a system of polarization and depolarization that is going to be occurring. Okay. And so that's that excitability. So it would be kind of nice next time I'll think about it. Have you ever, I didn't put batteries in yours, but have you ever licked a battery, like a 9-volt battery, put your tongue on it?
And maybe as a kid, yeah, I probably did.
Okay. Okay, that sensation there, right? That's an example of polarization, okay, or excitability, right? So you're polarizing your tongue in that case. Okay. Okay, so we know that our muscles, they're going to be elastic. And you guys know what elastic means? So they're going to have elasticity associated with it. What does that mean? I see yes. Stretchy. Okay. So they're going to have some stretchiness to it, right? Okay. And so because our muscles are going to have to be able to contract, you know, and extend. Okay. So
Sounds like pregnancy.
yes, during pregnancy, you use a lot of your muscles, right?
Mm-hmm.
Okay. So we're going to be talking a little bit about these topics. So contractility and extensibility. Okay. Okay, so let's start with the skeletal muscle. Come on. Come on. Okay, so in your skeletal muscle, there are going to be three connective tissue layers, right? So this here basically is going to be your bundle of your skeletal muscle. So, sorry, we have, so here's going to be your skeletal muscle. So we're going to have this very first bundle of muscle fibers, and they're going to be called the fascicles. Okay, so these guys are going to be covered by the perimysium, which is going to be let's see, where's my, okay, let me clear this. Okay, so this guy here, so as you can see here, this basically represents the muscle fascicles. So these guys are going to be covered by this perimysium. And then you also are going to have what we call the endomysium, which is going to be that internal layer. So those are just going to be tissues. So they're those muscle fibers. These muscle fibers are covered by tissues that are kind of collecting those muscle fibers, kind of organizing. Kind of like, so you have like a copper wire, and then that copper wire is covered by plastic that semi or non-conductive plastic, right? An insulation, right? An insulation plastic. And then you may have, let's say, if we're talking about wires for lights, right? You'll have your positive wire, you'll have your negative wire, and stuff like that. So how they're all insulated. So that's similar to what's happening with the muscles. Muscles have, they're kind of collected in fibers, muscle fibers bundled, okay? And then, so you have your perimysium, which is going to collect, it's going to collect the inner layer or the middle layer of these fibers. So we have these guys that are all collected. And then we have what we call the endofascicle. So the endofascicle is also going to be a subset that's going to be in between, right? And then we have the epimysium, and so that's going to be the one that's collecting the larger muscle fascicle. So if we take a look at it, here's a skeletal muscle. We have our epimysium, which is that outer layer of tissue, and then the inner part. These are your muscle fascials. So then those guys are collected by another layer that's surrounding that. And then if we go even further, so those guys are broken up into even smaller satellite, smaller bundles. And so, and then those guys are going to be covered by the Endomysium. And so if we get even those inner parts, so these are going to represent just your muscle fibers themselves. And then that is going to be covered by the sacroilium. And then you're going to have your myofibril. And so that's going to be your myofibril is composed of your actin myosin chains. Okay, so these guys are going to be broken down to even further. So the proteins of your actin and myosin. Okay. And I think I have a picture of that here in a second. So. Okay. Okay, so this here is basically a picture of your muscle fiber. So, and so you can see the filaments here. These are your myosins, and then over here the green parts. These are representing your actin filaments. Okay, so your thin little layers here, actin. Your thick purple layers here, that's going to be your myosin. Okay, and so you kind of see how it's integrated to each other. So you have what you call your M line meaning your myosin. So you have the chains of myosin that are wrapped around it. That you also have these the filaments themselves. Then you have what your I band, your A band, and then you have again your I band. Okay, and then you have your sarcoplasmic reticulum located at the end of that muscle fiber. And so if we take a look at the cells itself, so this basically is these rods here, the mitofibrils. So these guys are, again, they're just actin and myosin proteins, right? And there's some other proteins that'll be in there that kind of keeps them together. But for the most part, it's just going to be chains of actin and myosin. So those are just proteins themselves. So this here, this muscle fiber here is kind of representing a cell, essentially, right? So you have your mitochondria, you have the sarcomere. This is just basically your plasma membrane. Okay. So, and then remember with skeletal muscles, how it's multi-nucleated, right? So you can see your nucleus like towards the edges of that muscle fiber. So this muscle fiber is one entire whole cell, okay? And so we know that this guy is going to be stratolated in appearance, right? Because that cell itself is going to have that tone where it has those. Let's go back. So where you're going to have your nucleus on the edges, and then you're going to have that surface in the middle, right? So that striated appearance. And so we're going to actually see that appearance here a little clearer because of that result. But you can see that within that cell itself, we have the actin and myosin chains that are sitting there that are kind of controlling that muscle contraction, thanks to that extent. Okay. So let's talk about the sarcomere. So the sarcomere, like I said, is going to be composed of actin and myosin. So this is the structure down here. This is the myosin tail, and this is your myosin head. So myosin actually binds to actin. Actin is going to act like little chains for the myosin to come roll across. Okay. Now this process is regulated again by ATP and then also calcium. I said calcium formation is necessary. The calcium ion is necessary for muscle contraction. So your levels of calcium in your body is going to be that need to be controlled and regulated. So there's going to be a calcium homeostasis, which we've talked about before. You guys remember that?
Yes.
Okay. That's going to be regulated because the formation of myosin is dependent on it, okay? Or I should say not the formation, but the dimerization of myosin. And so if you actually look at this, the protein itself is actually composed of 2 myosin proteins. And so it's a dimer that forms. You have your myosin head that binds onto the actin chain. And so myosin rolls along those actin chains for that contraction. And so when we're trying to contract, you'll have myosin moving along the actin chain. And then when it extends, then it'll be let loose or it'll retract. Okay. Now, the actin molecule itself goes through a process of polymerization. So it polymerizes and depolymerizes. Okay, so that also helps with that chain formation and stuff like that. Because if it's not there, you can't have myosin moving along that chain. Does that make sense? And there's certain toxins and certain drugs that are used for causing actin formation, polymerization, and then also depolymerization, right? And so some of the things like in terms of muscle relaxers or things that could affect that part. So a lot of that has to do with messing with the actin and myosin chain itself. Okay. Okay. So these filaments here, like I said, they are composed of the actin subunits that are polymerizing. You have tryptophan, and then you have tryptomycin. Okay, so these are 2 other proteins that are involved in the formation of these filaments. Okay. So the sarcomere is just the connection of the actin and myosin chains in that organized fashion. So myosin acts like a motor, right? So, and actin kind of acts like, let's say, best way of saying this. So have you guys seen, people that, what are they called? The people that slide down mountains on a line, and they'll use the little wheel thing. Can't think of it now. I want to go. Let me.
Zip lining?
What was it? Zip lining. Thank you. Zip lining. Okay. I know it's a weird analogy. So the myosin, so you know, in case of the zip lining, you have the ones that are really fancy that have the wheels and everything like that that are associated with it. So. And so the person hangs on to that part. So that's kind of like myosin. Myosin is like that. And then the actin, or the actin filaments, right, are kind of like the zip line. Okay, so myosin acts like a little motor that sits there and pushes its way through that process. Okay, and so this process has to be, in a sense, kind of regulated. Okay, so that we can get that contraction and retraction, or contraction and extension. Okay, so the process of regulating that is going to be done by the nerves, right? The nerves kind of tell you what you need to do, right? Whether you should move your hand, whether you should flex your muscle, you know. So it needs some type of stimulus, a nerve impulse, right? And so let me see. We have what we call our neuro muscular junction. So neuro. Okay. So if you take a look at our image here, so this is our nerves. And remember, our nervous tissue is insulated, right, with the myelin sheets, right, that are surrounding, kind of acting as insulation so that those impulses aren't lost. And so these are your axon terminals. And so if you take a closer look at that, so you basically have your synaptic vesicles that are going to be releasing your acetyl-CoA, your acetylene choline, which is it's like a neurotransmitter, a signaling molecule. And then on the surface of the cells themselves, they have channels that allow, or receptors that will bind the acetylene choline. And these receptors that are binding the acetylcholine are also pumps, right? And so they're sodium ion channel pumps. So the binding of them allows that channel to open up. Okay. Are you with me so far? And so by the channel opening up, it causes a change in that charge state between the outside and the inside of the membrane, right? So and that should not be sodium minus, that should be sodium plus. So it allows the sodium ion to actually internalize. So then the sodium ion internalizes and it causes that depolarization. So your positive charges are able to go through that muscle fiber, and then it causes it to depolarize. Okay. Questions, concerns, Cash? Come on now. Sorry about that. Okay, so this is just another view, and then now we're introducing a couple of okay, so we're introducing some other structures. So we have the T tubule. So the T actually stands for transverse, and the T is so if you take a look at this guy here, the sarcoleum, you have so you have the T tubule, which is kind of connected to the sarcoleum. So this allows the pulse to be fed to be extended to the entire muscle. So it kind of like carries that charge, that electrical pulse, to everything else. So it allows the pulse to be distributed around the entire muscle fiber itself. Okay, and then you have so this guy distributes the pulse. The triad is just a combination of the it's going to be your sarcoplasmic reticulum, and it's going to be your tubule, and so it's just kind of like it's three membranes that kind of acts like it's three membranes that kind of work as a team. And so you have your pulse that is being distributed through here. You have your sarcoleum reticulum that is basically receiving that pulse, right? And so, and your and so you're getting that polarization that's being distributed along the membrane structure of that entire fiber. So this guy receives it, and these guys allow for the distribution of it, okay? But it takes the connection between your T-tubule in your triad formation here. So that band there represents that triad. Does that make sense? I'm trying to think of an analogy, but I can't think of one right now. Okay, it's like this. So you have electricity that is going in to your house, the electrical line that's going into your house, right? It comes in with one line, and that goes to your electrical box, you know, where it controls everything. So you have that one power line from the city that goes into your house. That goes into that box that controls everything, right? And so you have a little switch that turns off the lights in the kitchen and turns off the lights in the bathroom and turns off the light in there, right? Mm-hmm. So this guy here kind of represents the line that's coming inside, the direct line. This guy here, these guys, is that box that distributes the electricity to all of the different components in your house. Does that make sense?
Yes.
The middle one will be the support, and the other two next to the middle one are just helping spread the what it need to do.
Yeah, so this is kind of like your feet line. So this guy here is your feet line, right? And then this guy is what's receiving all of the pulses, right? So if we go back, you go here, and we have this nice little junction. Let's go back. Okay, so this is just showing you the little pulse, right? So this here is kind of like getting that pulse, right? So in the case of the T line is, come on now. So this is like a very local one, but what's happening is, sorry, I can't get back. So your T-tubule, so this guy here, that guy is providing each of those individual impulses, right? But it's a whole unit that's providing it. And then we go here, that just represents that membrane, right? That is providing it to. So this is that membrane that is being provided to, right? So your electrical pulse is being received here, and it's just conducting all of the electricity that is going through. And then that's being distributed by this guy, you know, so that the full fiber is being stimulated, not just that one part. Does that make sense? So it's being distributed through the link. Okay, so you're not acting only on like the end of the fiber, you're acting throughout the entire fiber. Okay, so it's just the conduction of those electrical pulses throughout the entire fiber, so you're stimulating the entire thing. Does that make sense? Yes, you're the only person I can see. If you don't say yes, nobody else. Okay, so let me see if I can give you like just a quick draw. Okay, so like I said, in the city has one line that goes to your house, right? And then the city has a line that goes to Jesse's house. This is Jesse's line. This is Letitia's line. Okay, now in Letitia's house, Letitia has lights in every room, right, Letitia?
Mm-hmm.
Okay, your stove probably uses electricity, your TV uses electricity, you know? So then it gets distributed to lights, refrigerator, TV, etc. Right? So in Jesse's house, same thing happens, right? It gets distributed. Okay, so this here represents Jesse's house represents one muscle fiber. This here represents, your house represents another muscle fiber. So what's happening here is that this is kind of serving as that line that's coming into your house. This is the line that's coming into Jesse's house. So this part is being distributed. You're getting those pulses being distributed there. This part, you're getting the pulses being distributed in there. And so it's making sure that the full, like both you and Jesse's house are able to get electricity, right? Mm-hmm. So if we screw up down here, if we break the impulse initially down here, then you guys don't do it. So like if you have an accident that messes up a nerve ending, then you screw up that nerve, you also screw up those muscles that are downstream of it. Does that make sense? So like somebody who is not able to move from their waist down because they screwed up their spine, right? They had a fracture in their spine, and so they're not able to move from the waist down. So that's an example where they've broken that nerve ending there, right? Let's say you have trouble with your arm, and so you broke your nerve ending in your arm. So then that is closer to the local area, right? So that's kind of like looking at that individual muscles problem. Or you have trouble moving a finger or something like that because you screwed up a finger, right? So you can narrow it down, but it's kind of branching in the system, in that system. Does that make sense? I feel like I'm just paddling there. Are you with me or are you guys not with me? Is it not clear?
Yes.
Okay. Okay, so let's move forward. Okay, so now remember we talked about muscle contraction, right? Okay, so now this process is going to be, it's going to be the forming of crosstalk between the actin and myosin heads that's going to cause that contraction, okay? So there are going to be a couple of things that need to happen for contractions to occur. One of those things is we have to have calcium, right? So calcium is going to be calcium initiating the contraction itself. Okay, so what's going to happen is that our nerve pulses is going to cause things to be excited. So we're going to get a shifting of the sodium ion entering the channel. That's going to cause depolarization, which is going to cause a change in that charge along the membrane surface. So you're going to get what they call an action potential traveling along the D line. So that action potential is then going to cause calcium to be taken up. So calcium is going to go inside. That calcium is then going to start interacting with the actin and the actin chain, actin and myosin chain. And so long as you have ATP, you're going to be able to sit there and get movement, right? So then the ATP is going to bind to the myosin head. Myosin is going to break ATP to ADP. And this is going to cause your muscle fibers to continue to shorten. And that processing of that ATP to ADP is going to continue to happen until you run out of ATP to sit there and cause that process to occur. And so in that case, the muscle shortens, we're going to get contraction, and then it's going to make tension. Okay, so this process is a signaling cascade. Have you guys heard of that before?
Mm.
Okay. So we have the active potential. I mean the action potential that's going to be delivered by the neuro nerve junction. Neuromuscle junction, and then we're going to have acetylcholine released. The acetylcholine is going to bind to the receptor. It's going to cause the sodium channel to open up. Sodium channel is going to cause depolarization of the membrane, which sends a signal down to allow calcium in so that we get calcium to start binding to the actin, myosin, and actually it binds directly to troponin. Then that's going to cause troponin, the polymerization of your actin chain. Your myosin is going to then start binding to the actin chain. As long as they're ATP, your myosin is going to move along that chain to lead to muscle contraction. Okay, so ACA binding to the receptor causes the internalization of sodium. Sodium then causes the membrane to be depolarized, which sends a signal to calcium to be uptook. Calcium binds to the troponin. The troponin then allows for the linking of the actin chain. The myosin heads move along those actin chains, causing contraction. Okay. Now, for relaxing of those muscle fibers, then calcium has to be removed from those structures, right? So then there's tryptomyosin. So the tryptomyosin basically reshields the binding sites that allows the tryptonin from binding with that calcium, right? So then calcium isn't no longer able to bind to it. You're not going to have any ATP. So you're going to run out, you've already run out of ATP, so you're no longer getting contraction. And then in this case, you kind of become fatigued. So as a result of that, no ATP, no binding of your myosin to your actin chains. And so your muscle starts to relax. Okay, now you'll also get actin depolymerization. So those actin chains will also be released as well. So you won't have these nice structural changes as well. Okay. Questions, concerns, Cash?
Nope.
Okay, so clear this up. Okay. Okay, so here's your myosin. These are your actin filaments. Okay, so during muscle contraction, your myosin is going to be binding to your actin filaments, and it's going to be moving along those cables, right? It's been moving down, so it's going to cause it to contract, right? So you're getting that pulling in. Okay, now when you run out of ATP, then your myosins are going to let go, and then you're going to get retraction or extension, okay? And it's going to relax. Can you guys see that? Okay, so again, this is just the more elaborate details of what's going on for the muscle contraction. So on the active sites, you're going to basically get binding of calcium to the troponin. So calcium binds to this part. This allows the myosin heads to start to attach, and then myosin binds to the actin. It moves on the, so myosin itself, the myosin head has an actin binding site. And so it forms like a little bridge between the two, and it starts moving along that bridge. So as we get the release of ATP, as ATP is being converted to ADP, so you get the breaking down of one of the phosphates, one of the inorganic phosphates, it's released. Then another, it binds another ATP, it goes through that process again. Okay. And so, and this guy will continue as long as we have ATP present, right? As soon as we start running out of ATP, then we're going to get total release of the myosin chain. Okay. So, and this guy will continue moving along that pathway long as you have ATP present. Okay. Are you with me? So we talked about hydrolysis before, right? And I should probably bring this up. So the terminology of ATP going to ADP in an inorganic phosphate is basically, it's being hydrolyzed. Hydrolyzed basically means you're adding water, and that causes to break things up, right? So you're using water to sit there and break the bond between the last phosphate, because ATP is basically adenine triphosphate. So there's 3 phosphates on there. So we'll put an A with a circle, and then there's a phosphate there, there's a phosphate here, and a phosphate here. Okay, so those P's represents the 3 phosphate. It's your PO4 molecules, right? So, and okay, so we break one of those by adding water, and we get 2 P's, right, for phosphate groups. And then we'll get this PI by itself. That PI represents inorganic phosphate, which is just this molecule. Okay. Questions, concerns, Cash? Okay. So you know an old typewriter, right? Have you guys seen an old typewriter where you hit a key?
Yes.
Okay, and it shifts over, right?
Yes.
Okay, so that's kind of like what's happening with the myosin head. It binds ATP, and it processes ATP, and it causes it to shift. So you get that first movement. Then it binds ATP, it reattaches, it processes ATP, it shifts over another. So it's like hitting the next key, hitting the next key. Okay.
Almost like that little duck, or that one where you put, I think it was like water, or it had some kind of like liquid, and it just like tips down.
Yeah. 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 It is like that. Okay. Okay, and oops, got carried away. So in terms of muscle metabolism, so you guys heard of creatine?
Mm.
Yeah, no, maybe. Yeah. Okay. Well, creatine is it's a protein that is it basically will bind ATP, right? It causes the processing of ATP, meaning that it will actually take ATP, and then it'll take one of the phosphates from that ATP and keep it on itself. And then as a result of it, you get ADP. So then you have what we call the creatine kinase. It'll actually remove that phosphate from creatine, and then it'll actually put it back on ATP, okay? And then you get creatine removed. So it goes through this process all over again. So when the muscle is relaxing, we take the ATP, it binds to creatine. Creatine then is phosphorylated, and it has a phosphate on it. And then we have the creatine kinase, which removes which is weird. It basically removes, it transfers the phosphate from the creatine back to ATP. Okay, so what's happening in this situation is that creatine is kind of being used as an energy source, right? And then it's going to be recycled, go through that process again. So in the case of water, so these are the places where we're going to get our ATP from. So the first one is that we're going to be recycling that ATP using creatine. The other places that we're going to be getting it from glucose, right? So either from our blood sugar or from glycogen. And then here during glycolysis, we get the production of a little bit of ATP, right? And then we get pyruvate. Well, if there's an aerobic respiration, we're going to end up with 32 ATP. Which is shown down here. Aerobic basically means that there's oxygen present, and then it goes through this process here, okay? The pyruvate does. Now, if there's no oxygen, we get lactic acid buildup. Lactic acid goes into the blood, we get cramping, okay? If we were yeast, we'd make alcohol. Okay. So here, during cellular respiration, we have that pyruvate becomes pyruvic acid. It gets processed long as there's oxygen present, and we get 30, as I said, 32 ATP, right? If it's blood sugar, it's technically 30, it's a total of 32 ATP at the end of the day. If it's blood sugar, there's that 2 additional, but they get used in this process. I should actually say this pyruvic acid, it's going to be 30 ATP is going to be generated. If it's blood sugar, you get the 2 that's added on through the process of the glycolysis. And then fatty acids can also sit there and provide ATP through it, but it just depends how much it provides. Which stage you use the process. Okay. So 3 ways of getting it. We can get it through creatine, which is just kind of like it's slowly transferring the ATP. It's like a recycling of that ATP. We have the first one will be through glycolysis. And then we have the option of going through which cellular respiration via the mitochondria. Now, cellular respiration in reality combines both this process, this early stage of glycosylation, glycolysis, as well as the cellular respiration in the mitochondria. Okay, this part here happens in the cytoplasm. And that one happens in the mitochondria directly. Okay. Okay, so questions, concerns, Cav? Okay, So aerobic respiration is what? Aerobic meaning what? Anyone? Anyone? Breathing. Okay. Oxygen. Yes. So aerobic respiration is when there's oxygen present. Anaerobic respiration when there's no oxygen. Okay. Are we good? So anaerobic respiration only leads to 2 ATP being generated. Aerobic respiration leads to a lot more, tons, 32. 32 from a single sugar molecule. Okay, so let's talk about muscle contraction. Oops, I carried away here. Okay, so there are different types of muscle contractions. One type is isotonic muscle contractions. So isotonic basically means well, there's two types of isotonic muscle contraction. There is concentric and there is eccentric. Okay, and you can kind of see concentric. It's being shown here. So when we have that movement, as in movement going up, concentric, okay, and then eccentric is we have the movement going down. So we're muscling up, flexing. So we get contraction, elongation, contraction, elongation. So contraction, concentric, elongation, eccentric. So the other type of muscle contraction is isometric. Isometric basically means no movement, so we're just leaving our arm up, you know. And so in that case, it's still, we have contraction going on with the muscle, but it's being evenly, you know, it's being even out. So isometric, okay? So isotonic, two types, one contracting, concentric, one elongating, okay? Question, concern, and?
Mhm.
Okay. All right. Okay. Okay, so this is just basically showing you the ideal length of the sarcomere. And so this here represents so if we have the maximum length where there's no overlap between those actin filaments, the thick and the thin filaments, thick being the myosin and the thin being the actin. So no overlap at all. So no cross bridging. That's what's being shown here. This guy is considered increased length. So where you're just getting the ends of those actin filaments just to start to match up with the ends of the myosin filament. And then this guy is considered decreased length when you've gone beyond those ends, okay? And so during maximum tension, being 100% tension, look at that, that's about, so we get that 80 to about 110, roughly. And so, or it says actually 80 to 120. I don't know about that, but it's more like 110 to me. But 80 to 1, according to the book, 120 is where we're going to get the maximum tension occurring. Okay, so when we're at the max tense tension, it's going to be between that decreased length and that increased length, like the lower half of the increased length. So we're going to have that little bit of overlap. So the frequency of frequency is, it deals with the tension over time. Okay, so we have 3 different phases. We have the latent phase, we have the contraction phase, and we have the relaxed phase. Okay. Latent phase, it's basically is that very first phase where it's right before you're getting ready to contract, right? So, and so you're basically going from that relaxed state to a contracted state. So the time that it takes us to get there is, like 5 milliseconds from a latent stage to start going through contraction, right? That contract period basically goes from 5 milliseconds to about 26, 27 max, right? Is our contract phase. And then from our contract phase to relax. So to get kind of completely relaxed, it's going to take us about 90 milliseconds. Okay. So in other words, it's going to happen pretty fast. So that process of that calcium ions binding to the troponin, and then that process is going to take us to get that contraction, that complete contraction. It's going to take us about what, 15 milliseconds or so. So the process is pretty fast, in other words. So we respond pretty fast to that first stimulus, right? Because we're on the milliseconds, on a milliseconds frequency, right? Does that make sense? That we respond, well, I guess it does make sense. Otherwise, you would move, right? You put your finger on something, you would move, or anything. Right? Let's say you smash your hand, or your hand is smashed on the thing. That stimulus, because you're going to contract or something to move back from whatever you put your hand under, right? Your hand gets caught in a door or something like that. Don't you respond almost immediately, pretty fast?
Yes. Yes.
Okay, so this basically is So this first one is called the wave summation. So basically you can see that the wave is being, you're getting a consistent stimulation cycle. You're going up, you're going down, you're going up, you're going down, you're going up, you're going down. And so it's going to be like an additive effect that's happening, right? And then at the end of it, you're getting released, right? So in other words, what's happening is that you're getting excitation, contraction, excitation, contraction, excitation, contraction, excitation, contraction. And so it's going to be like an additive effect of stimulations, right? And so in that sense, it's all being added. Where the second one, this guy is called tetanus. In this case, you're getting the stimulation. Stimulation is so high, right? You're getting so much stimulation that you basically have gotten rid of your relaxed phase. And so you've overextended that stimulation, you're overly stimulated, you're leveling off, and then it basically, contraction is going to continue, right? And so you're going to get a totally continuous stimulation, right? So you're at a high, and that's it. Okay. So this guy is your summation, wave summation, where stimulate it, you're allowed to relax a little bit, you stimulate it, but you're not allowed to relax completely. Does that make sense? You're not able to totally relax under these circumstances, where this guy is, you're stimulated so high, you reach your peak, and that's it. And you're just there, continuously. Okay. So your muscle, right? So have you guys ever had a muscle twitch?
Yeah.
So where it's like, and it keeps, your muscle keeps just going like that?
Yeah.
Okay, so that's kind of like your wave summation, right? Where you're just getting that muscle twitch happening, right? So you get that first one, you get the second one, you get the third one, right? Actually, the next one.
It happens a lot once in a while in the eye. Why is that? I mean, you look like a little
No, I know exactly what you're saying. And I don't know why it happens a lot in the eye, but you're right, it does happen a lot in the eye, because I know my eye tends to do that frequently.
And then people think you're winking at them.
Yeah. I don't have a good logical reason for that, right? So, but in this case, you're looking at it, so your twitch initially starts off light, then it gets a little stronger, and then a little stronger, and then a little stronger, and then a little stronger. So that's kind of like your wave summation, your wave summation curve here. And then your tetanus one is, like I said, you're just overstimulated. It's like, bam. That is possible because we know that sodium is one of the things that depolarizes and polarizes the membrane, right? So that is possible. Okay. So there's another process. It's called trepi, and it's like a staircase where you're actually getting stimulation, a little stimulation, and it drops all the way down. Relax. Then a little stimulation, relax. A little stimulation, relax. Right? A little stimulation and relax. Okay. So in that case, yeah, it's just up and down, right? So a little bit of stimulation, you relax completely. You go up a little bit more, then relax completely. So it's kind of like a staircase of stimulation, right? But it's not, it's more gradual, but you're going completely down compared to when we talk about wave summation, right? So in the case of tetanus, I should also mention that we shoot up all the way. So basically you've opened all those channels. The calcium is just, it's saturated with calcium. And then it stays until you basically get muscle fatigue and you're not able to produce anything more. So basically you ran out of ATP, you have nothing else, and then it drops down. Okay. Let's see, yeah, come on now. Okay, so in the case of marathon runners, so a lot of distance marathon runners, so they're trained for those long distances, right? And so if we sit there and we think about what's going on under their circumstances. Okay, 1 second. So like marathon runners are kind of trained so that their muscle development. So often marathon runners are often very lean, things like that. But how they train their body by doing that those marathons is like they kind of have to work up to it, right? And so, but their muscle fibers are also going to be a little bit different. Let's say a trained marathon runner going to be a little bit different than, let's say, your normal Joe that's walking down the street, right? And this. And so we have these. I mean, what? The fibers that are going to be generated are going to be a little bit different, and the ratio that you're going to have of the different fibers are going to be different in somebody who is like stationary, who sits around all day at a desk like myself, right? Compared to somebody who is athletic and is constantly moving around and constantly doing things, right? So I am at a loss for words because I don't know what SO stands for. I have to get back, but on that. So, but it's noted that they have, there we go, slow oxidant. Okay, thank you. Look at you, good job, thank you. So, in this case, since we know that oxygen is important, Long distance runners, basically, they have a larger number of slow oxidative fibers, and then that's relative to what they see in terms of the few of them that are fast oxidative. So in other words, that the fibers are able to sit there and, I guess, maintain their oxidative state longer than the fast fibers. And okay, so then in the situation that in that case, they're going to use what they call a slow twitch, right? So it's going to be more like so when we look at that frequency of contraction and everything like that. So let me see, hold on. So in their situation, it's going to be kind of like that wave summation for that slow oxidative state. Where in my situation, it'll be more like the tension, right? So where I'm going to shoot, like if I try to run a marathon, I'm going to shoot my load fast, and then I'm just going to crash, right? And so I won't make it in that case. Where a person who runs frequently is going to be more, you know, up, down, gradually doing that summation. Okay. Let's see. So where, let's say, a bodybuilder is going to have a lot more of the FG fibers, and then they're going to have fewer of the fast oxidated, the FOs and the SO fibers. Okay, and so in this situation, they're going to have bigger, bulky muscles ultimately, where the runners have the nice, lean muscles. So bodybuilder is also going to be considered a hypertrophy. So because hyper meaning more, greater. So in that case, they're going to be producing more muscle cells, and those muscle cells are going to be bigger in size, bigger in growth. Atrophy is basically when you lose muscle, right? And so like if you're not using muscle, like me, I go through atrophy. So the difference between a sprinter and a long distance runner, question mark. So the difference between a sprinter and a long distance runner, sprinters typically, they're gonna have more of the FG fibers than a long distance runner. So typically their legs are gonna be much bigger, big thighs, big muscles, so they're going to have a lot more hypertrophy, and then they're going to have more hypertrophy. And so, but they're going to have a lot more of the FG fibers than the FG fibers, and also the FO fibers, right? The fast oxidative fibers compared to a sprinter, where a sprinter is going to have the SO fibers, more SO fibers. Okay. Okay. So cardiomuscle tissue, we know that that guy's only found in the heart. So the way that it's organized is let's see. Okay, so we know that it has a single nucleus, and we have the muscle fibers itself. Here's a cardiomuscle fiber. It has that single nucleus, and then we have what we call gap junctions. In our gap junctions, we have our desmosomes. We have desmosomes. We have these gap junctions, and then we have down here we have the actin-myosin chains. So the gaps between those muscles, these are called, they're going to be intercalated disc. And so basically this is going to be the sarcomere, and then also the gap junctions between the two muscle cells. So this is one muscle cell, one muscle fiber. This is another muscle. Okay. And,
Dr. Henry, it's already 11:10.
okay, I'm over. So it's time for,
Oh.
time for lab. So I'll meet you guys in lab.
Yeah.
Sorry about that. I only have like 3 more slides, but we'll finish it on Wednesday. So I'll meet you guys in lab.